all cells in an individual will contain almost the same information, is what we can gather. And there are, again, variations to this, but this is genetically, this is what makes us very much so alike, even though we're from maybe perhaps different backgrounds or even regions of sorts. When I was talking about the cell division process, I mentioned something called caretaker genes. Now, this is not the same as housekeeping genes. With housekeeping genes, this more so relates to the transcription process during more of a cell replication component of sorts, not necessarily cell repair within the cell cycle. So even though caretaker and housekeeping might sound the same, they're kind of functionally worlds apart. All right, here's kind of where I want you to pay a little bit more close attention: genomic imprinting. So as far as the transcription process, and this is going to be something that we would want to occur because we need DNA replication and so forth in order for us to exhibit certain traits and inherit certain, genes and traits. Now, if we have something called genomic imprinting present, this means that we have transcriptionally kind of inactivated a certain copy of the chromosome. An example here is listed. A copy inherited from father is active, while a copy inherited from mother is inactive or silenced. Kind of back to the topic and concept from slide number 1, methylation. If we turn off a gene, this basically renders it silenced. It's not going to express itself. And that has a lot to do with, again, how methylated these particular genes become, the cytosine end of things. And that will affect, again, if we have a gene that is silenced. So genomic imprinting and gene silencing kind of go hand in hand in that sense. Transcriptionally silenced genes are said to be imprinted. That's where that type of statement comes from. So this doesn't necessarily mean this is a bad thing. What it means is we either turn off the paternal component of a gene or a maternal component of a gene. And that just means we inherit something different. Maybe we don't inherit something from dad, but it comes from mom instead. And that really, in the big picture of things, doesn't make things such a big deal, unless mother really contributed or father contributed something to us that ultimately ended up with the offspring having some type of disorder that was debilitating, I guess we could say. But otherwise, it's not really too big of a deal to silence particular genes. It's a normal process. With the genomic imprinting, in fact, we can do this and describe it in a number of manners. So we have something called biallelic expression and monoallelic expression. So if we're looking at biallelic expression, this means that we inherit genes from maternal and paternal sources. If gene silencing occurs, more than likely this is going to inactivate maternal or paternal genes and traits. So with monoallelic expression, that means we get randomly chosen inactivation in some form from one parent versus another. So here we have maternal copy randomly chosen for inactivation in some cells, paternal copy in others. This is going to allow us for the combination of maternal and paternal different expression that we could see genotypically and phenotypically in many cases. If we looked 100% like father, maybe internally we're like 100% 100% like mother. No, that's not always the case. But there are more so random selections of genes that are chosen that never get expressed while others do. So it makes us unique in that sense. We're a different recipe of our parents of sorts. There's a mentionable here that I should bring up, genetic conflict hypothesis. So this says imprinting of maternally inherited genes tends to reduce offspring size. Imprinting of paternal inherited genes tends to increase offspring size. Now this is more or less still being researched and tracked rather. But it's a hypothesis. Of course, hypotheses are just kind of questions that remain to be researched with full widespread answers. If that's the case, I guess we could look at our own heights and kind of determine if that's true. Maybe if we have siblings, this might be a little bit easier to follow.
I have a question. Yes. What do you mean by size? What context is that? Like the height, or what do you mean by size?
Mostly it'd be in the concept of height.
Okay.
Yeah. But again, it's more or less a hypothesis of sorts to spark conversation. If anything, spark conversation, because it's not any type of completely proven item at all. One of the things that I would want you to know for the test is distinguishing Prader-Willi and Angelman syndromes. Realistically, they are the same thing, with the exception of the presence of genomic imprinting, the silencing of maternal or paternal components. So, for example, we could have basically the same description with really one minor change, giving us either Prader-Willi or Angelman syndrome. What they both share is over 4 million base pair deletions. It's going to be on the long arm of chromosome 15. So chromosome 15, if we're looking at the karyotype chart, it's going to look at the upper larger portion of, again, chromosome 15. So if we delete 4 million base pairs, this still means basically a lot of different instructions. What it can end up with and manifest as would be a couple different things. There could be short stature involved. Both of them kind of have a hand at variable mental disabilities in cognitive development. So there could be, again, variable cognitive disability there. Both also kind of lead to the susceptibility of obesity. But mostly what they share is, again, that deletion of material. 4 million base pairs in the long arm of chromosome 15. Except, this is what kind of distinguishes them. It comes from the father's side, or paternal side. It would lead to Prader-Willi syndrome. If it comes from the maternal side, it would lead to Angelman or Angelman syndrome. And this is one versus the other. Some similarities. A lot of times with these particular patients, there's also growth developments that also are exhibited with some of these children, especially notable in childhood. So if we're deleting from the long arms, we have Prader-Willi to the A on the A side, and then Angelman to the right on slide B.
So just to clarify, you did say that they both would be carriers of the chromosome 15, correct?
It would affect the long arm of chromosome 15. 4 million, yeah, 4 million, the deletion of 4 million base pairs of chromosome 15. What this would have to deal with in relation to gene silencing is it would be on the paternal end of things. If we have effects on the paternal side of things.
Then it would be the Praters, correct?
Right, Praters, yeah, Prader-Willi. On the maternal end of things, it would be Angelman or Engelmann syndrome. Sometimes how I remember them would be trying to just kind of find silly ways. I think of Willie as a guy's name. I have a friend named Willie. Maybe that's why I think about that. And then I know several female colleagues named Angel. And I think angelic as maybe a feminine role of sorts. So somebody that's if you're associating kind of like how I was in my head remembering these, Angel, think female, think maternal, Willie, more of a masculine of sorts connotation. So I would associate that with the paternal side of things. Although if there are any ladies out there that have a nickname Willie, that kind of goes out the window. But as far as just kind of silly ways of remembering them, that might be a little helpful. Prader-Willi, paternal side of things, causes the deletion of 4 million base pairs of chromosome 15. Angelman would be the maternal side, deletion of 4 million base pairs, still of chromosome 15. Slight variations that lead to very similar conditions. Then we have Beckwith-Weidman syndrome. This one's a little bit more so related to an overexpression of another component of the body. This is IGF-2, or active insulin-like growth factor 2. I just wanted to bring this one up. I'm not necessarily testing you on this one. Brings it because it's a very characteristic condition. You see kind of the larger tongue, and that's a shared similarity with those with this condition. Larger tongues. Oftentimes there's a gastrointestinal, as you can see, kind of a protuberance of sorts within the abdomen. And then there's a characteristic ear fold that we would see. It's going to be more so on an ultrasound if we're doing like a ultrasound wellness baby check, or maybe just kind of just noticing it. And of course, B would be, now we notice it on the infant, but the ear fold is going to be creased characteristically in the way you see it there. There's another condition or syndrome, Russell-Silver syndrome. I don't have any pictures for this one, but this is a downregulation similar to Beckwith-Weidman syndrome, but downregulating active insulin growth factor 2. It causes more so diminished growth in those that do survive after this condition is present. As far as epigenetics and nutrition, there is a relationship between nutrition deprivation and disease. I think you guys would probably note this widely with all types of patients that you might have interacted with at one point. With nutrition deprivation, this can affect food and electrolyte balance, and really what we're looking at is obtaining minerals, electrolytes, and all sorts of different elements to help kind of keep us in balance. Electrically, especially when we're dealing with certain charges within the body. The body especially likes potassium and sodium for the purpose of cell functioning. Sodium-potassium pump, you might remember. And we'll go over that too during the next couple of weeks. And then also, as far as electrical charges go, the relationship that the body has with potassium and calcium. Something known as the resting membrane potential that kind of helps drive a lot of our all types of different neuromuscular functioning within the body. There could be alterations to methylation, which ends up kind of explaining why exposure to stress early in life changes our behavior in adulthood. And these stresses early in age can be from different health conditions and just kind of how the body reacts to certain treatments if they're rendered early in age. But overall, it would take kind of a very unique cases and special cases early in life to really shorten the lifespan as we go into adulthood. Here's another syndrome that is still in our population, fetal alcohol syndrome, and this mostly deals with the effects of ethanol or drinking alcohol during pregnancy and how it could affect children, not only cognitively, but also give us more of a phenotypical kind of hallmark appearance that is notable, likely with the ear positioning and then also eye folds. So a slew of different syndromes. Fragile X site, I'm not going to test you on. This one is a really unique form of mutation, but more than likely, this is something that you will rarely ever see in many acute care settings or hospital settings. There might be some folks that may have had Fragile X syndrome that have made it past adulthood. It's one of those syndromes that, again, we rarely see, but still can occur. Obviously, it's documented as part of some of the different traits and disorders, syndromes that exist as part of the database we discussed earlier. I don't have any pictures to show you for FSHD. I think pictures would tell a whole lot more related to these particular issues. If we're looking at a review of the topic of genomic imprinting, remember genomic imprinting, we're looking at kind of the shut-off switch, so to speak, for certain genes. So given that, if we're looking to define it or describe it, which one of these would you think? Chat box says C. Genomic imprinting, I mentioned earlier, goes hand in hand with gene silencing. So the answer would be C. Is anyone here a twin by chance? Oh, we have twin. Okay, sorry. I had to look at the expanded box. All right, as far as twin, are you an identical twin or
Maternal.
Okay. I guess this would deal with identical twin studies, but mostly these are going to be studies that look at kind of the effects of different manipulations within the environment and maybe even methylation patterns, many of them being environmental in nature, that could cause notable changes between cells that under the microscope technically are identical. So these are, there's a lot of insight that can be gathered by looking at twins and some of these different manipulations. There have been kind of instances and cases where that have been followed where maybe twins not separated at birth, perhaps, or not in the sense of keeping the same parent, but maybe adopted identical twins, just kind of growing up a little bit differently. And some of these patterns that they noted were just kind of based on environmental changes that they went through. So they didn't grow up obviously in the same environment. So they were separated by adoption. And later in life, kind of just looking at some of those changes, although they take identical twins and will take a look at some of the studies, some of the similarities that they share and some of the sequences that might be different based on different activities. Maybe one smokes, maybe one doesn't. So of course, to take part in any type of study, somebody has to consent to it, and in particular, when looking to recruit participants, they're looking very specifically for identical twins to continue the education and insight as to how different environments do have an influence on DNA sequence changes. Epigenetics in cancer, I'm going to go to this screen. If we have enough of a manipulation of DNA, what can arise are different gene mutations. There are certain markers now that we have understood that can be identified that identify some forms of cancer, not all, but some forms. And oftentimes, the ones that we do know of, we're really looking and testing folks to see if we could proactively care for these particular patients and prevent any future probable health discrepancies or ailments. So what we're looking at are certain maybe tumor markers, perhaps. It might be present. Retinoblastoma 1 gene is one of them, and there's a RAC1 and 2 that could be inherited, related to some breast cancers and maybe tying with reproductive type cancers as well. So these are things that, again, we can look at and identify early on in many cases, especially if it does tend to run in the family, and see if there could be any proactive treatments to prevent something that possibly could be inevitable, such as acquiring certain types of cancers. This particular section I'll go into when we talk about the cancer section. I brought it up here mostly because it's applicable to epigenetics, but again, it kind of deals with different opportunities for mutations to occur, because when we're looking to replicate, there has to be some form of stop, kind of like tumor suppressor gene stops for and checks for mutations